You're on mute.
Okay, hi. I'm gonna start again. Basically, I'm working off a PowerPoint, so if you ask a question, I might not be able to see it in the chat. So feel free to just like unmute yourself and ask. You can ask before, during, after, anytime. So this is called Intro to Nucleotides. So it's mostly going to be about that. And then all of the information that I use is taken from the textbook, which is The Molecules of Life, and then also from the lecture notes. So refer to like those or the textbook if you have any other questions. And just like a general tip I have is that the textbook like really helped me get through the class. And like whenever I was questioning something, I just went to the textbook. And since it's clearly outlined in the lecture, like it follows really well. So that would be like general advice. Just reading, especially the parts you don't understand. Okay. Can you guys see? There's like, sorry. For some reason it's not. Okay, so what does a nucleotide consist of? A nucleotide consists of a pentose sugar, which is seen here, which is the 5-membered ring, attached to a nitrogenous base, which can be seen at that side, and a phosphate group, which is linked here, or it can be more phosphate groups. So the sugar contains 5 carbon atoms and is a cyclized form of a pentose. The carbons are labeled C1' to C5', starting with the carbon at the glycosidic bond. You can just memorize this, but it's going to be kind of useful to know the numbering of the carbons because a lot of times someone will reference it as like a substituent of the C2 carbon or the C3 carbon. So that's something important to remember. So when you look at this molecule, do you guys know, like, will this be found in DNA or RNA? Does anyone know?
It would be DNA because the C2 prime is a hydrogen.
Yeah, okay, awesome. So it will be DNA, and you can tell that by looking at the C2 prime carbon because DNA is deoxyribo, and basically that indicates that at the C2 prime there is no oxygen group. So ribonucleotides, which are found in RNA, are derived from ribose, which is a pentose sugar, which has a hydroxyl groups attached to the C1', the C2', the C3', and the C5' carbons. And in ribonucleotides, the difference is that the C1' OH group is replaced by a base, and so that's where it like attaches to like the traditional bases, and then the C5' OH group is replaced by a phosphate group. But you can see that there's still an OH on the 2' hydroxyl. And then the deoxyribonucleotide is, so the nucleotides that make up this DNA are deoxyribonucleotides, and they're not derived from the ribose, but they're instead derived from the 2' deoxyribose, in which the 2' OH group in ribose is replaced by a hydrogen. So that's just like the easy way to tell whether it's DNA. And it's like very important to recognize that there's a difference in composition, even though it seems like a really small molecule, like one small change. The difference in composition of deoxyribonucleotides and ribonucleotides helps explain the different properties of RNA and DNA that you've already been learning about, like especially like folding and stuff, and that you'll be learning about soon. So about the phosphate group, so it's attached to the C5' carbon, and it can have either 1 or 2 additional phosphate groups added to it. In terms of nomenclature, nucleotides with 1 will be mono, 2 di, and 3 triphosphates, which makes sense. And then the phosphate groups are called the alpha, beta, and gamma phosphates, alpha referring to the one closest, and then beta, and then gamma. Again, these things are just like kind of good to memorize because it'll help you like envision things when people are talking about them. So for nomenclature again. So the combination of sugar covalently bonded to the base is called a nucleoside, and then the combination of the nucleoside plus the phosphate group put together is called the nucleotide. So obviously these are really similar words, but again, it's good to just kind of memorize because if if you're taking a test, like someone could throw one word in instead of the other and it might change like the answer completely. So make sure that you're looking out for like small things like that. So the nucleotide bases in RNA and DNA are just substituted forms of two heterocyclic molecules known as pyrimidine and purine. So pyrimidine has a single six-membered aromatic ring with two nitrogens and four carbons. And the nitrogens are at the 1 and 3 positions. So that's how the numbering will work: 1, 2, 3, 4, 5, 6 for pyrimidines. And then purine has two fused rings, one with 5 atoms and one with 6. And in the purine, the nitrogens are at positions 1, 3, and 7 and 9. So this is a little bit like more complicated, but you can kind of figure it out. And then, so the sugar in the pyrimidine is attached to the N1, and then in the purine, it will be attached to the N9. It's important to memorize the difference between a purine and a pyrimidine, and just like memorizing these general structures, although it seems like kind of unnecessary, will make drawing the actual bases, the substitute forms, a lot easier. And the way I remember it is just that pyrimidine is the longer word, but like the smaller molecule, and purine is the smaller word, but the larger molecule. So kind of like an opposite thing. So DNA and RNA, as I'm sure you know, have different bases. They each have two purines and two pyrimidines. The DNA bases are adenine, guanine, thymine, and cytosine, whereas the RNA is adenine, uracil, guanine, and cytosine. So thymine and uracil replace each other in DNA and RNA, but they're very similar molecules, just one has an extra methyl group. So it's important to know about the different bases because each base has a unique pattern of hydrogen bond donors and acceptors, which allows DNA and RNA to serve as templates for the transfer of genetic information. Basically, each base contains like unique sequences that allow unique patterns to be transferred. So another important part of this class, and also a lot of MCB classes you'll take in the future, is being able to identify hydrogen bond donors and acceptors. So the red arrows are pointing towards hydrogen bond acceptors, and then the blue arrows are pointing away from hydrogen bond donors. And then, so another question you may have is how like are these molecules formed? Because they're so like large initially, you might like not know how they're formed, but in both DNA and RNA, nucleotides are joined together by the formation of a phosphodiester linkage between the 3' carbon of one nucleotide and the 5' carbon of another. And so there's kind of like a phosphate group like in the middle that's bridging the two nucleotides. So the phosphorus atoms forms two ester bonds with the oxygen bonded to the 3' carbon of the first nucleotide, and then another oxygen bonded to the 5' carbon of the second nucleotide. And this creates like the backbone of either the DNA or RNA molecule, which is some terminology that people use pretty frequently. And it's important to note that the phosphate groups are negatively charged, which is a very important determinant of the three-dimensional structures formed by DNA and RNA, because as you'll learn, like the different structures are kind of composed the way they are because of the need to like the negative charges repel each other, so that they have to be like far enough apart. And with regards to synthesis, so synthesis of both DNA and RNA is a stepwise addition, and it So it's adding nucleotides to one end of the chain. The 3' OH group attacks at the end of the chain attacks the alpha phosphate group of an incoming nucleotide triphosphate, which releases a pyrophosphate, which can be seen on the right side of the picture, which is two phosphate groups connected. And the way that this reaction works is that the triphosphate group is really high in energy, and its hydrolysis drives the reaction. Okay, and that's all that I have. Does anyone have any questions? If not, then um,
Are you going to be going over fixed law later?
I'm sorry, wait, I couldn't hear that.
I just asked if you were going to go over fixed law.
I won't be. I don't think anyone else is covering it, but I'm not sure. Okay, so I just saw the chat, sorry, but I can.
Sorry, I didn't understand what the question was. What law? Can you hear me, Heather?
Oh, I fix law. I'm not actually sure what that is.
Fixed law is not a part of this course. Why are you? This is Sarah. Why are you asking about fixed law? Fixed law has to do with viscosity and diffusion of different liquids past each other. Something we're going to cover in this course. Is there some confusion? Now, it is something that's in the textbook, but it's in like chapter 16 or 17, and we just don't get to it. I don't know if they cover fixed law in 100B, but it's not this course.
Okay, so the next tutor can go, but I can post my slides on the Piazza page after this.
All right, can everyone hear me and see my screen?
Yeah, you're good.
Awesome. All right, hi everyone, my name is Chetan, and I will be talking to you about DNA structure, specifically base pairing and the double helix. So let's talk about base pairing first. So important rule of base pairing, you always have one purine paired with one pyrimidine, as Heather talked about. So you got your purines over here, A and G, paired with always pyrimidine, C or T. You can also have uracil as well. And I put down some mnemonics there if that helps you memorize them. There are plenty of mnemonics online if you want to search them up as well. But important thing for DNA, one purine, one pyridine. And because of that, that leads us to Chargaff's rule. I don't think you guys covered that yet. I assume you'll be covering it a little bit later. Basically, the amount of adenine in a DNA molecule will equal the amount of thymine, and same thing for cytosine and guanine. You can do some pretty basic math with that. I'm sure you'll have some problems about that later, if not already. And important thing to note is differences in the number of hydrogen bonds between these nucleotides. When we look at AT, AU, you'll have only 2 hydrogen bonds. And note the placement, which molecules are actually involved, or which atoms are involved in those hydrogen bonds. And for cytosine guanine, you have 3 hydrogen bonds. So I just have a quick question here. In theory, based on the number of hydrogen bonds, which is more stable, DNA molecule with high GC content or high AT content? Anyone wants to unmute themselves and just
High CG, high CG content.
High CG content because
it has more hydrogen bonds, it's more stable.
Yep. And we'll actually talk more about stabilization and what a very important factor is base stacking in a little bit. But yes, high GC content would be more stable. But before that, let's talk about the double helix in general. So it is a right-handed helix, and I know this is something I struggled with, and many of my friends who took the class struggled with, is how can you tell if something's right-handed or left-handed? And this is going to be pretty important for DNA, and also when you study alpha helices a little later on in the course. I'll try and make this easier for you, but really it's something you have to work out yourself. If we take, let's say in this DNA molecule, this orange strand right here, if you can take your right hand and you can kind of wrap your hand around it and follow it along with the inside of your palm here, between your thumb and other fingers, that'll mean it's right-handed. If you can do it with your left hand, it's a left-handed helix. So just practice that by yourself and eventually you'll pick up how to tell the difference. And of course, like Heather talked about, phosphodiester linkages link these. Here's the reaction summarized right here. 3' OH, tax phosphate, 5' O, 5' carbon. And these strands are also anti-parallel. So important thing to note, they're anti-parallel, run in opposite directions. So you can see here, this runs purple strand 5' down to 3'. And the orange strand go moving in the same direction, runs, has the opposite, 3' down to 5'. And just in terms of structure, you have the sugar phosphate groups on the outside of this, and they are negatively charged phosphates. And those negatively charged phosphates can, they may repel each other slightly. And they can also interact with, water or maybe some positive cation, whatever may be near the protein. That can provide some stabilization from that repulsion as well. And then the base pairs are on the inside. Sugar phosphate groups outside the backbone, base pairs on the inside. Remember your base pairing rules. And finally, we'll talk about base stacking. I didn't see this in your slides, so you may be learning this later. But I'll try and make this simple because this was also a little confusing when I learned it. But each base will have partial charges, right? We can see there's some electronegative atoms in each base. You have your oxygen, nitrogen. So like this oxygen will be pulling electron density, nitrogen will be pulling some electron density. They'll be partially negative, possibly. And these carbons, where they're attached to, these will be partially positive. So that's what I mean by each base has those partial charges. Some parts positive, some parts are negative. And when you rotate those bases on each other, there will be favorable overlap between the partial positive and negative and vice versa, between the top base and bottom base. And that's what this gray shading is symbolizing. Sorry, it's not the greatest picture. I made these at short notice and it was the best thing I could find. But basically, these things will overlap and that's favorable overlap and will help with the stabilization of the DNA helix. That's really important.
Question.
Yeah.
A base stacking, is it basically a dipole-dipole interaction between the bases? Or is it like a van der Waal interaction?
Well, I was going to talk about that. Yeah, they're van der Waal. Oh, someone going to say something? No, okay. Yeah, so there is definitely van der Waal interactions, of course, those temporary dipoles. There will be that part of the interaction. I believe this can also be electrostatic, if I'm not mistaken, and dipole-dipole as well. So really a big mix of all those interactions contributing to base stacking. Or I should say base stacking contributing to stabilization. And remember, just note for van der Waals are very weak interactions, but when you add them all up together, they will have a significant effect adding to stabilization. And that's really all I have to cover for my section. Does anyone have any questions about base stacking or anything else I talked about previously?
I had a question on the overlapping partial charges.
Yep.
So when you have the overlapping base pairs like you have in the picture, will it always overlap favorably like that, no matter the pair?
So I can't tell you too much because I'm not too sure myself, but I know depending on which pair or which bases are on top of each other, you'll have different parts overlapping with each other. So if you had like 2 purines or 2 pyrimidines, because they're the same, you may have more favorable overlap, or you may have different overlap than say a purine and a pyrimidine on top of each other. But I don't know, I couldn't tell you specifically which one is more favorable, which one's not more favorable.
Okay, thank you.
Yeah. Mm-hmm.
I also had a question.
Yeah. Yeah.
Does the stacking also contribute to like the rotation of it? Does it cause that, or does something else cause the rotation?
I think it's a combination of this so this will stabilize that rotation, but there's also, like the negative phosphates on the outside. Those may repel each other and contribute to that. So there's a lot of different factors contributing to that rotation and stabilization. Yeah.
Okay, thank you.
Mm-hmm. Anyone else?
I have another question.
Mm-hmm.
Yeah, so by saying favorable overlapping, what do you mean by that? Like, is the partial positive part corresponds like stack with the partial negative?
Yeah, so like if you see where my pointer is, pretend this is a partial positive carbon. It's over this partial negative nitrogen. So that's what I mean. Partial positive near partial negative, that type of stabilization.
Okay, thank you.
Mm-hmm. Anyone else? Okay, I guess we can move on to the next tutor if no one has any questions. Feel free to put them in the chat as well. And I can also post these slides once we figure out where is the best place to put them.
Okay, so next I'll be talking about how the intermolecular forces that you've talked about a little bit in this class overall contribute to nucleic acid structure, specifically in the context of forming the DNA double helix. So, okay, I see. So I want to motivate these slides by first talking about, why does a DNA double helix form? Because so far we've seen what the structure of the DNA double helix is like, what the Watson-Crick base pairing that goes into it is, but we haven't really talked about the energetics of why it happens. So I want everyone to think about the formation of a DNA double helix as an equilibrium process where you have reactants, which are the single-stranded pieces of DNA, eventually coming together, doing the Watson-Crick base pairing and forming the double-stranded helix. And from observations, we know that if you look at lots of living cells, for the vast majority of cases, DNA is always found in the double helix form unless the cell is dividing or unless a specific gene is being transcribed. So from that, we can see that this equilibrium between the single-stranded DNA versus the double-stranded DNA, it heavily favors the products. So that kind of suggests that there must be some amount of energy favorability. There must be some amount of increased stability in the DNA double helix that's causing this to happen. So next, I want to talk about what are some of the factors that could be contributing to that. So does anyone want to take a guess as to what factors might lead to increased DNA stability? I think we've talked about it a little bit so far. Anyone can chime in.
Hydrogen bonding.
Yes, hydrogen bonding is one. Any others?
Base stacking.
Yes, base stacking. So I think we've hit the two major ones right here. So hydrogen bonding and definitely base stacking. Another one is cation interactions with a phosphate backbone, which I think hasn't really been introduced yet, but I'm sure will be covered in lectures that are following, but this will be a little bit of a preview. So now we've talked about factors that contribute to increased stability, but can anyone think of any reasons as to why the DNA double helix might be less stable? So thinking in terms of entropy, is there a penalty to forming a helix?
Size.
What was that?
Size, like how long the strand is.
Yeah, that definitely contributes to it. So specifically, like this was mentioned at the end of Tuesday's lecture, in which as you get a longer and longer DNA double helix, then that increases or that decreases the entropy, or it decreases the amount of disorder. And so that leads to an energy penalty in terms of entropy loss. So next, I want to talk about how these forces eventually add up and lead to the equilibrium that we see here. So the first one force that I want to talk about is hydrogen bonding. So this is what your DNA double helix and its single-stranded pairs look like before the helix forms if you're in an aqueous environment. So notice that on the left-hand side, before the double helix even forms, you have your nucleotides such as cytosine and guanine that you can see here, and all of their hydrogen bond acceptors and donors are already engaged in hydrogen bonding because they're surrounded by water all the time. And so even after the DNA double helix forms, it looks like the hydrogen bonds that were previously there when the single strands were in water are now, so those hydrogen bonds with water are lost, but they're essentially just replaced with the hydrogen bonds that the nucleotides make with their complementary pairs. And so what this tells us is that if hydrogen bonding is essentially on both sides of the equilibrium, it kind of cancels out when it comes to the energy contribution to stability. So essentially the gain in stability from hydrogen bonding is almost negligible, because if you have hydrogen bonding before DNA double helix formation and also after it, it's not the main driver of the DNA helix forming. However, hydrogen bonding still serves a pretty important purpose in terms of forming the DNA double helix. Does anyone want to guess as to why hydrogen bonding is still important, even though it's kind of necessary but insufficient? Okay, so the main reason why hydrogen bonding is still quite relevant for the formation of the DNA double helix is that it kind of enforces the fidelity in Watson-Crick base pairing. So for example, you see that the hydrogen bonds that are lost are immediately recovered, but only if the Watson-Crick base pairs form. So for instance, if you had cytosine pairing up with adenine here, you would see that this hydrogen bond donor on this amine group would be lined up with the hydrogen bond amine donor on the adenine, and then that would not be a stable hydrogen bond. So only when you have Watson-Crick base pairing can you recover the hydrogen bonds, and only in that case would you have a kind of zero loss in energy as you move from a single-stranded DNA to double-stranded helical DNA. And one question that I kind of want to leave all of you with and may come up in future assessments is what would happen if you had DNA double helix formation in a hydrophobic solvent in which you don't have access to hydrogen bonds before the double helix forms, and what if you had DNA double helix formation in a vacuum? Then what becomes the role of hydrogen bonding in kind of driving that process? So something that you can think about or ask on Piazza. Any questions before I move on? Okay, so the next force that I want to talk about is cations and the influence of cations on enforcing stability of the DNA double helix. So here's a structure of DNA. This also comes from your textbook. That's what TMOL is abbreviated as. And as you can see from the backbone that we've talked about so far in today's concept session, it's very heavy in terms of the phosphates that are there because every single nucleotide has its own phosphate group, and that contributes to a pretty high density of negative charge that's all packed into one structure if you have the DNA double helix forming. And so whenever you have the accumulation of all these like charges together, that creates a repulsive force that could potentially destabilize the structure. So one way that you could deal with this destabilizing force would be to introduce the opposite charge, because then the opposite charge would kind of induce an attractive force, first of all, and then it would also screen the negative charges in between each other. And so typically what happens is magnesium is the divalent cation that serves this role. So notice that magnesium has two positive charges in addition to instead of just having one. So you could do bi with relatively little magnesium and still kind of get the job done. So here's a schematic from your textbook of what that interaction would look like if you have an aqueous environment and you have two different charges interacting. And you might think from this diagram that the most favorable energetically would be panel C because it looks like the two charges are closest together. And in physics, you've learned that if you have two opposite charges and you bring them as close to each other as possible, then you have the most stable formation. But actually, it's panel B, or part B of this figure, that shows the most stable formation of the two ions. And the reason for that is when you're bringing the two charges close together, in addition to thinking about the increase in stability of bringing those two charges together, you also have to think about what's happening to the water molecules. So it's not just the ion pair force that's being involved here, but it's also the ion to dipole interaction between the positive charge in the water molecule and also the negative charge in the water molecule. So in the extreme case in panel C, when you have the two ions very close to each other, most of the water molecules are pushed out of the way, and so they lose out on a lot of ion to dipole forces, and that's energetically costly. So the optimum is actually B, which is the kind of hexahydrated form that you might see if you refer to your textbook. And to see kind of tangibly what this would look like if you have magnesium interacting with the DNA backbone, it would look somewhat like this. So here you have one phosphate group and another, and your magnesium cation is kind of screening the charge between the two. Any questions so far?
So for B, could you say it's more stable because of van der Waals also, because you're talking about like dipole, or is it not like temporary or?
Yeah, so someone was going to say something? Okay, so the reason why B is the most stable, so typically when you're asking reasons for like why it's most stable, you typically pick the strongest force. So even though, van der Waals is definitely playing a role in all of these, the most significant force would be the ion to dipole. So notice that the positive charge here is kind of like a permanent charge, a permanent dipole, as you said. whereas the water molecule is kind of a little bit more transient, but it's still a polar molecule. So when you have a fixed charge compared to a polar molecule, that can still induce a pretty strong attraction that's on a level that's not the same as hydrogen bonding, for instance. So it's a little level higher. Okay, any other questions? Okay, so now I'll move on to base stacking, which is one of the most significant forces in driving the stability of the DNA double helix formation. So here, these are maps of the electron densities of all the DNA bases. So as you can see, these DNA bases have regions of both low electron density and high electron density, which contributes to the character of being polarizable. But also, if you notice, every single one of these bases has at least one aromatic ring. And as a result of that, you have all these pi electrons that are delocalized. So if you'll recall from organic chemistry, if you just have a system that doesn't have any pi delocalization, the electrons that are surrounding an atom kind of more or less have to stay there. But if you have pi delocalization, then that allows the electrons to kind of move around a lot more. And that contributes to more of a polarizable character, meaning that if you brought a negative charge closer to one of these molecules, then the electron density could shift away in a way to sort of handle that. And I actually have the same exact figure that Chetan had, but here I can annotate it in a way that I think was actually very helpful for me in terms of visualizing this when I was learning about it. So if you look at the bottom part of the molecule, the guanine, because this carbon is surrounded by 3 electronegative nitrogens that are more electron withdrawing than it is, you might end up with a region that's slightly electropositive here, whereas this nitrogen in the cytosine that is relatively electronegative compared to its neighbors has higher electron density. So that could contribute to the electrostatic component, at least, of base stacking. And throughout this molecule, you could also have van der Waals interactions and other dipole-dipole forces that contribute to base stacking being very energetically favorable. And the key takeaway is that this is the driving force in causing the DNA helix to form, because this is the greatest contribution to the increased stability of the DNA helix relative to the single strands. Any questions so far? Okay, and so far we've listed a few forces that increase the stability of the DNA helix. But now I want to step back and kind of think about, is there anything that might be destabilizing the DNA helix? So in other words, are there any forces that actually favor the formation, or at least favor single-stranded DNA instead of double-stranded DNA? And so here we can talk about entropy. So both in the polymerization of DNA and also in the broad picture of the DNA double helix forming, we see that in DNA polymerization, as you're going from monomers to a larger polymer, you're decreasing the amount of disorder that's possible. So individually, your monomer units, your nucleotides, are free to kind of move around in whatever way. But once they've been synthesized onto the DNA, they're kind of in a very fixed orientation. And similarly, if you look at your single-stranded DNA, this one single strand is actually very flexible, and it's like a chain, so it can kind of move in very many conformations. But once you form your DNA double helix, then it's kind of locked into that conformation. And so it's a lot more ordered. And so in the process of DNA double helix formation, although there are gains in terms of base stacking, there's also a trade-off. You're also forming more and more ordered structures. And as a result of forming those structures, there's a sort of like an energy penalty that offsets. And so I think this is a pretty important question that I would like to leave you all with, which is, what are sort of the biological consequences of having a DNA double helix being too stable? Because if we ignored the entropy for a second and only looked at base stacking and the effect of cations and the effect of hydrogen bonds, then we would kind of think that the longer your DNA molecule gets, the more base stacking you have and the more stable it gets. But in reality, that's not the case. And you should think about what would happen if we ignored entropy for a second and had a case in which the DNA double helix formation was so product heavy that your DNA double helix was never able to go back to the single-stranded form. So that's all I have today. Does anyone have any questions? Okay, I'm going to pause my screen sharing and I'll post these slides in case anyone asks that.
Okay, can everyone see my paper well enough?
Yes.
Yes, okay. Okay, so I'm going to be going over the standard structures for DNA, which are B form and A form respectively, and then the differences in each of their ribose rings that causes these two forms. So here I have the ribose ring with the carbons labeled 1, 2, 3, 4, and 5 prime. This one has no hydroxyl group at the end, so this one will be DNA. And then this one does have a hydroxyl group at the 2' carbon, so this one will be. And if all 5 atoms in this ring are planar, if they're all in the same plane, then you're going to have some steric clash from the substituents because they'll be in eclipse conformations. So the way that the molecule is formed is the energy is lower when one atom is non-planar, so it pops out of the ring, and this will decrease the steric clashes. So here I have the ribose ring drawn again, and this in the drawing, this 2' carbon is coming out of the plane of the page towards on the same side as the 5' carbon, and so we call this C2' endo. So it'll be on the same side as the 5' carbon. And if it's on the other side of the plane, so underneath the ring, you're going to have it C2' exo. Okay. Over here, we have the ring drawn again, and then this time the C3' carbon is coming out of the page, so we would call that C3' endo because it's on the same side as the 5', and then same thing again, you're going to have C3' exo if it's underneath the plane here. And this is important because DNA is the only one that can take the C2' endo form. It typically doesn't take the C2' exo form. And this is because that RNA has the 2' hydroxyl group. And so if RNA were to adopt the C2' endo form, then you would have steric clash between the 2' OH and the 3' phosphate. So only DNA here, and RNA will adopt this form. And these sugar forms are important because this will dictate different helical structures for DNA and RNA. So another note on these sugars is which side the base is pointing towards. So here's the hydrogen, and then I'll draw a base here. So this is carbon 7, carbon 8, and carbon 9, or sorry, these two are nitrogens. But so this will be called the anti-conformation because the 8 on the ring and the hydrogen on the 1' carbon are going to be trans. And the other form is when you have, okay, I might I might run out of room here. So here you're going to have 7, 8, 9, and then these are going to be cis. Okay, and then this one is called syn. Yeah, a lot of this stuff I think you're going to be going over in lecture next time. Does anyone have questions on this? Okay. Oh, and this is because you can rotate about the glycosidic bond here. And the transform will generally be favored because there's less steric hindrance, but there are some cases where this form is favorable, like in zDNA, for example. Okay. The different structures now. So I'll start with bDNA, and here I've drawn a helix. So each turn of the helix, so this will be one turn. This is 34 angstroms, and then here will be the major groove, and then here will be the groove. And I've also drawn a base pair here, so here's the hydrogen bonding. This will be adenine, this will be thymine, and so on this base pair, you can visualize the major groove as this one over here, and then this will be the, sorry, this will be the minor groove. Okay, and this side is the convex edge, and then this one will be concave. And in the DNA, the edges of the base pairs are accessible because the bDNA major groove, so this side's pretty accessible because the major groove is wider and you can access these functional groups. So this will be a hydrogen bond donor, this will be an acceptor, this will be a methyl group, and then this is a acceptor as well, acceptor. Does anyone have an idea on why this might be important, as for each base pair there is a different pattern here? Anyone have any ideas?
It allows protein interactions for transcriptional factors, or it just allows protein specific interaction.
Yeah, great. That's exactly right. So yeah, regulatory proteins, you can fit an alpha helix nicely in here, for example. Yeah, and this will all be interacting with the edges of the base pairs. In contrast, okay, this one is accessible and wide. And then this is narrow and less accessible. Yeah. So another thing with bDNA is that it has a very uniform structure. AT, TA, CG, and GC are all about the same width, which is 11 angstroms. Okay, for this. And the base pairs also all have a similar shape. And this is important for things like DNA replication, so that, for example, the DNA polymerase knows the base pairs are correct because all the base pairs fit the same shape in the active site. Okay. So going back to the uniform structure of bDNA, you're going to have all the base pairs centered on the helix axis, and they're all going to be perpendicular to the axis. So this can be the axis right here. So here's one base pair, here's another base pair, and then here's another base pair. So it'll all be around here. And if you look at it from above, these will be the phosphate groups on the outside, and then in the center, you're going to have all the base pairs crossing in here. Okay, and so the electrostatic repulsion of the charged oxygens in the phosphodiester bond is what's giving it this helical twist. Okay, so does anyone have questions on bDNA? So I'm going to move on to A form then. A form, if we remember, will be with the C3 prime endo sugars. A form is for, it can be for RNA, it can be for RNA DNA hybrids. It can also be for DNA in some conditions, but generally bDNA will be more favorable in normal conditions. So, so these are, I would draw it, but I think that's testing my artistic abilities, but yeah, I think I forgot to mention, yeah, the helix is right-handed, like you heard from the other tutors. A form is also right-handed, but there are a few differences, like the base pairs are tilted away from the center. And also the in A-form DNA, there's less rise per base pair. So it's not going to be the 34 angstrom rise like in B-form DNA. And so that means there's going to be closer base stacking. So this will X diameter because the backbone negative charges have to stay away from each other. And so the result of this is that the base pairs are offset from the center of the helix. So if I were to draw it, it would look more like, so if this is the axis here, it would look something like this. Okay, and in this one, this will be the major groove. And then this one will be the minor groove. And in A-form DNA, you're going to have a deeper, narrower major groove, and it's buried in the helix. And so this means that it's going to be less accessible than the B-form major groove. And so there's a lot of consequences for that. And then for the minor groove, it's wide and shallow. Yeah. So that's mostly what I have for A-form DNA. Does anyone have questions?
I had a quick question.
Mm-hmm.
When does A-form DNA usually occur?
So it'll occur for RNA, RNA, DNA duplexes, and DNA. DNA is for only certain situations, so usually DNA will be B form.
When you mean certain situations, is it just like different, like environment, like environment where there's not a lot of water, or?
Yeah, if it's dehydrated, it might take A form because it'll be more stable. But if you have an RNA duplex, it's always going to be A form because it can't adopt the C2 prime endo conformation, which is necessary for B form because of that steric clash between the hydroxyl group and the phosphate backbones.
Okay, thank you.
Yeah.
Move your paper a little bit up so that we can see the whole diagram at the bottom.
Oh yeah, sorry.
Thank you. It's okay.
Okay, so I'm going to go ahead and stop the recording for this session.